As our last example of immunological methods, we're going to be looking at what is referred to as ELISA, Enzyme-Linked Immunoabsorbent Assay. Okay. If you go and you look, far left there is an example of these ELISA. Everything's done in one little well. Each of those wells, depending upon the size of the plate, what they're doing, if it's the 96 well plate, each well can hold 200 microliters, basically 2 drops of liquid. They also have 364 plates. 364 wells, each well can hold about 20 microliters, or a fifth of a drop of water. So what you're thinking? So what can be done here? Well, if you look in your textbook, where you can see a bigger picture of this, you'll see what they do is they take this special type of plastic that these well plates are made of, called well plates because it has a bunch of wells. Each well will be a different patient, or they can do multiples, 3 wells, 5 wells for each patient, so that way they have redundancy. They can line the well with either proteins taken from a specific microbe or with antibodies that are towards a specific microbe. They take the patient sample, they put it into the well. It will interact, bind to whatever was in the patient sample. If you're testing for the presence of a microbe in patient samples, you will line the well with antibodies that are specific for that bacteria or that virus. Patient sample you put in, if the bacteria, the virus is present, it will bind to the antibodies that are lining the well. Then you come in and you rinse away stuff, but the microbe is still bound to the antibodies that are bound to the well. You come in with other reagents that will react to the presence of the bacteria, the virus being there. Different methods to doing it, what they call indirect or direct. Are you testing directly towards something, or are you looking for the presence of, serum, or are you going to do multiple steps of, here's horse or rabbit antibodies binding to something, and then you come in with horse against rabbit, and unless you're planning on doing research, just know there'll be different steps. From a clinician standpoint, do you have to know whether it's horse antibodies, rabbit antibodies? No, because what they're going to tell you to do, here's the kit. Take the kit, open the kit up, make sure all the wells are wet with deionized water, dump out the water, because basically you're just rehydrating whatever the manufacturer lined the wells with. Add patient sample, serum, whatever wells that have been designated. Let it sit, incubate for so long, dump out or aspirate out. Add in 1-2 drops from bottle A. Let that sit, dump out. Add in drops from B, blah, 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 dump out. Add in final coloration solution, and then as it shows at the bottom there, look for yellow versus white, red versus clear, blue, whatever. So that's from the clinician standpoint. It's not going to be, is this horse? Do I need rabbit, anti-horse, things like that? You don't need to worry about that. Now, when you're looking at these immunological tests, you can take it further. You can do in vivo testing. You're looking for in the sample itself. You haven't isolated anything. Or you're looking at in a slide, a tissue slide. So you don't always have to isolate with pure cultures. You can do it in the mixed culture. You can do it in the tissue still that's been isolated, taken from a patient. As I've mentioned in a couple of the previous immunological methods lectures here for section 4 of chapter 15, these are highly specific, okay? The specificity could be high or low depending upon what you're testing for. Highly specific, you're looking just for E. coli 0157H7. Or you can lower the specificity, and we're just testing to see if this is E. coli or not. Sensitivity is such that, depending upon, are you doing a glass slide and you're looking with a microscope, or are you doing a Western blot, or are you testing for antibody titers? Sensitivity, you can raise and lower it depending upon what your threshold is. Turns out there's always going to be bacteria present in urine, just one of those things. But if you're doing one of these immunological tests for the presence of bacteria, how sensitive do you want this to be before it gives you a yes? Set the sensitivity low, you're going to get a false no. Set it too high, you're going to get a false yes. So you can gauge your sensitivity based on what it is you're trying to test for.